Okay, the second type of regulatory gene in cells are what are called proto-oncogenes. And RAS genes are an example of proto-oncogenes. Proto-oncogenes are going to be almost the opposite of our tumor suppressors. They are normally turned off. And when you're in the off position, you do no cell division. However, if you get a signal, remember signal transduction? This will turn on our proto-oncogene, and when it does, this allows cell division to proceed. So how does this go about? How does this RAS gene go about turning on cell division? So typically, we're going to start with a signal transduction gene. This is something that's happening up in the brain. These are your growth hormones. So these are going to typically produce proteins that will then travel through the body. So this will get transcribed, and it will then get translated, and we will create a protein signal. So here's our signal. Transduction is when it causes that. Like I say, a lot of times these are going to be hormones. Your growth hormones are going to tell your cells to undergo mitosis. So this signal, once it's created, will travel through the circulatory system, and it will find its way to a cell. So there's our typical cell. And on the outside of your cells, remember, you have receptor proteins. These are looking for very specific signals. So this signal will find its respective receptor cell, and it will fit into that receptor cell just so. Once it does, it will change this protein, and it will cause that signal to be transduced across the membrane. So as a protein, so as proteins change conformation, that's the fancy word for shape, this transduces the signal. So transduce is just a fancy way of saying that this message on the outside is carried across the cell membrane to the inside. So once we get there, this is where the RAS gene comes into play. You got to start though with that signal on the outside. So once we get our signal, I'm drawing a bigger cell here. So there's my cell wall. Here is my nucleus that has my genome. So here was my receptor protein. And here is that signal. The RAS gene produces a RAS protein. So on the inside, you have this RAS protein. So when this comes into this cup, it doesn't actually enter the cell, it changes the conformation of this receptor protein, which now turns on my RAS protein. So this here in green, this is my RAS protein. Now the RAS protein, what it does is it now starts a cascade of chemical reactions. So this will then cause a chemical reaction to produce another product, which will cause another reaction to cause another product, which will cause another reaction inside to cause another product. And then inside of the nucleus, this particular product will then activate the growth genes in your cell. So the RAS gene, once it's turned on, creates a cascade of chemical reactions. The final product will then turn on a particular set of genes involved in cell growth. As soon as the RAS gene starts this, it will automatically go back to the off position. If it doesn't automatically go back to the off position, we're going to keep this cascade going. We would get cell growth, cell growth, cell growth. So the important part of these kinds of regulatory is as soon as this turns on, as soon as it starts that cascade, it goes right back to the off position. We have no more signal here, and so it'll stay into the off position until something else falls into that pathway. This is where the problem comes in. The mutated form doesn't allow it to turn off. If I have a mutated RAS, so we'll make a mutated RAS. So here is my mutated RAS protein, mutated. A mutated RAS protein doesn't actually need a cell signal to get turned on and begin this cascade. A mutated RAS is always in the on position. Remember, these proteins are supposed to be in the off position most of the time, so that they only activate one signal and then they go back to being off. As soon as you have a mutated RAS, you no longer need to be induced by a signal. You are in the always on position, which tells the cell to keep dividing. And this is where that mutation leads to uncontrolled growth. Mutated RAS is always on, therefore we get uncontrolled growth.